Hi everybody! Today I'm going to be taking my bee traps out for the very first time. I'm super excited, so I'm going to be taking y'all with me while I go look for some trees to put them in. I get so distracted out here on the trails. There's leafcutter ants, army ants, spider monkeys. Lots of people study different ways to sample bees, but it's usually through traps placed on the ground. A lot of these are modified versions of pan traps, or bee bowls if you're cute. This is a new design of bee trap that I've created to sample the Canopy bees here, and I'm hoping it's going to work. There are only a few studies that have surveyed Canopy bees using baits like this. Some are in temperate, cooler North America, and some in tropical Africa, and none in this region. I did some research and talked to a few of the scientists that have been working with these canopy traps and created this design of my own. The main piece is a clear disc of plastic that holds the cups and allows them to be suspended up into the canopy. This allows me to leave the trap in the tree and just take out the cups to get the samples out, and it makes all the cups level with one another to avoid any sample bias from passing bees. The entire thing gets held with washers to a tree line and suspended up into the canopy, and each cup has water with just a little bit of soap in it. The soap isn't to clean the bees, it's actually to break the surface tension of the water. Since insects are so small, physics doesn't work the same way for them as it does for you and me. Water is much thicker in a way, and if they land on water, a lot of the times they'll just float on top without breaking the bond of the water molecules. This is good news for bees, but bad news for bee traps since it means that they can just crawl out of the trap easily. A tiny bit of soap works to remove that surface tension so I can sample the bees properly. So let's talk about the cups themselves. These are clear solo cups that have been spray painted with white, fluorescent blue, and fluorescent yellow. These three are the three standard colors for bee bowls. We use different colors because different bees are attracted to different colors of flower. And we use fluorescent paint because bees, unlike humans, can see into the UV spectrum, which gives them really cool access to colors that we can't see with the naked eye. Fluorescent paint shows up differently under a UV light spectrum than normal paint does, and it can make these traps more attractive to bees. Some bees prefer blue or purple flowers, some prefer yellow or orange, and others prefer white. Flower color is just one of the many ways that flowers can attract pollinators. This starts to tap into the very cool study of what are called pollination syndromes, or how there are different groups of flowering traits that have evolved to attract different groups of pollinators. For example, a red flower with a long tube usually attracts a hummingbird. White flowers that smell very strongly and open at night tend to attract moths and bats. And some flowers, like orchids, go absolutely wild with scent and mimicry and patterns and traps of their own to lure in pollinators. Since I'm not trying to catch hummingbirds or butterflies or anything besides bees really, I'm going to stick to blue, yellow, and white since I know that bees like those. I'll have to change the design if I start to catch anything on accident, like frogs or other vertebrates. I'm also going to try putting a few holes in the cups. When it rains here, it pours, and I don't want my cups to overflow, so I'm going to try to poke some holes where I want the water line to stop. I'm doing all of this because the pollinator network, or the things that pollinate and all their flowers, is a really important web of interactions in a given habitat. One of the reasons I'm trying to study arboreal canopy bees is because things happen quite differently in the canopy than they do on the ground. Especially in a rainforest like this, we're working in a much more three-dimensional habitat space. Here on the Osa Peninsula, trees can regularly get over 30 meters or over 100 feet tall. And what this means is there are a lot of living and physical factors that make the environment very different, especially for something small like an insect. For example, the canopy gets a lot more sunlight. I've just pulled down this tracer line, and this was its original color. It was red. And this is the rope that I pulled down from up above. So it's the same rope, but you can see it's gotten completely bleached out by the sun. There's also a lot more rain that reaches the upper canopy compared to the understory. What this difference in water and light availability means is that most of the flowering plants here are actually the trees themselves. If you look around at eye level, it's actually very green and it's hard to find flowers here. This is super different from the desert southwest of the United States, where I've done most of my other research, where most of the plants are just a couple of centimeters high or some chest-high flowering shrubs. So this is all super new and exciting for me because it's a totally new way for me to look at flowering plants and bee activity. So I'm trying to understand the spatial organization, basically the heights where the bees are hanging out here in this very tall tropical rainforest. Now that I've got my first bee traps up, I just have to wait and see what they catch. In the meantime, I'll be climbing up in the canopy myself to catch them in action. Thanks for listening. I'll see you all soon.